overview of one way to clone genes, to move them into a bacterium, for example, E. coli, and make copies. So to give you some direction with regard to what I'd like you to know, on D12, under the Chapter 8 module, way down at the very end of the module, I just posted a practice Chapter 8 mutations quiz, and Chapter 8 mutations and Chapter 9 recombinant DNA quiz. So that would be an idea of the specifics of the chapter number information I'd like you to know. Okay, so we will assign that as homework. If we have a little bit of spare time right before the lecture exam 3, we'll go back and review this, but we are running a little bit behind, so I'm going to assign it initially as homework. And then today, after finishing Chapter 8, we're going to move on to Chapter 13, viruses, which is, I think, you'll find fairly interesting. And just for those of you that might have time over spring break, Professor Nagamura just introduced me to this incredible book. It's by Nathan Wolfe. He did his undergraduate at Stanford, his graduate degree at Harvard, and he's one of the young up-and-coming virologists that's working on predicting viral pandemics. So anyway, I just started reading it last night, and it's like a novel. It's so cool. It's called Viral Storm by Nathan Wolfe. So just wanted to give that a little promotion there. Okay, so, and the last thing is, we will have a short open lab tomorrow, 8 to 9:30. You want to remember the first lab back after break is lab practical 2. So tomorrow will be the last time to review.
You said 8 to 9?
Oh, excuse me. I'm sorry. 9 to 10:30. I apologize. 9 to 10:30. I'm sorry. Oh my gosh, give me another cup of coffee. Okay, 9 to 10:30. Sorry. Okay. So what we're going to do then is we're just very quickly going to finish off Chapter 8. You guys will recall that we finished last time a discussion of transduction, which was the horizontal transfer of donor bacterial DNA to a recipient by a bacterial virus, a bacteriophage. And we had used our discussion to understand how transduction can lead to the evolution of new emerging bacterial pathogens. We used the example of the Shiga toxin producing E. coli 057H7. And we described how it appears that a bacteriophage transferred the Shiga toxin gene from Shigella dysenteria to E. coli. And hence, we ended up with a brand new toxin-producing strain of E. coli. So, trying to answer the question, well, why do we care? Why should we learn about transduction? So from there, we're just going to finish the last few topics. And one of the last topics here is on plasmids. Plasmids, you guys will remember, are described as extra-chromosomal. That means they're outside the chromosome. Double-stranded DNA, circular double-stranded DNA. And these plasmids usually carry extra genetic information. So plasmids are classified according to the genes that they carry. So we can have, for example, conjugated plasmids. And conjugated plasmids, if you're talking about gram-negative bacteria, they would carry the information for formation of a sex phyllis, also called the conjugation phyllis. And would also carry the genes that would permit the donor bacterium to transfer a copy of the conjugated plasmid to a recipient bacterium. Assimilation plasmids, you guys were going to skip, so you don't need to worry about them. But R factors is a generic term for DNA which encodes DNA sequences which encode information for either antibiotic resistance and or heavy metal resistance. And just to describe why we often see genes that permit these bacteria to grow in the presence of heavy metals. Prior to the discovery of antibiotics in the old days, often the only treatment that was available to help patients that were infected with bacterial pathogens was the use of heavy metals like silver or mercury. And you might recall that from Chapter 2, we said that heavy metals can cause protein denaturation. So this is probably the mechanism by which they could inhibit the growth of pathogenic microbes. But the problem is heavy metals are totally non-specific. So not only will they cause denaturation of the microbes' proteins, they would cause denaturation of the patient's proteins as well. So they tend to be very, very, very toxic. But it appears since those heavy metals were used for many, many years, there was natural selection for bacteria that evolved the genes to be able to detoxify the heavy metals. And thus we often find on these ARC resistance factors, we'll find genes for antibiotic resistance, and often we'll find resistance for heavy metal genes, heavy metals. One of the common genes that's found is for an enzyme called mercury reductase. It helps to detoxify mercury, which has historically been used as an antimicrobial. So these R factors, they may be found on plasmid, in which case the plasmid would be classified as a resistance plasmid or an R plasmid. And what's very disturbing is that these R plasmids may carry genes for resistance against multiple classes of antibiotics. So an R plasmid may carry resistance genes for 4, 5, 6 different classes of antibiotics. And even more disturbing is often you get a combination. You'll have the conjugated plasmid carrying some of these R factors. So you have a conjugated resistance plasmid, and that's really disturbing because it means that the host bacterium can share copies of that R plasmid, and again, the R plasmid may carry genes for resistance against multiple classes of antibiotics. That donor bacterium can share copies of that R plasmid with many, many neighbors. And so we would see the rapid spread of antibiotic resistance if we had a donor bacterium that had a conjugative resistance plasmid. So this is just an example of an R plasmid. So we see here that these little abbreviations are the abbreviations for the antibiotic resistance gene. So here's a gene for tetracycline resistance, chloramphenicol resistance, streptomycin resistance, which is an aminoglycoside, sulfa drug resistance, and this is the mercury reductase, an example of resistance genes against one of the heavy metals. Origin of replication, plasmids are often self-replicating, so they can make multiple copies of themselves, and this would be the origin of replication, just as we saw in the bacterial chromosome. And then here, this is a conjugated plasmid. So here is the origin of transfer. This would be the site at which a copy of this plasmid would be transferred to a recipient bacterium. So again, in one transfer event, we would be transferring resistance to 1, 2, 3, 4 different antibiotics plus a heavy metal. Yeah, so these things are not good, not good, not good. And then a second type of genetic element that bacteria can share are called transposons. And transposons are really fascinating. They were discovered by a Cornell geneticist by the name of Barbara McClintock. Transposons have been nicknamed jumping genes because they can move from chromosome to plasmid, they can move from plasmid to chromosome, they can move from one place in the chromosome to another, so it almost seems like they're jumping around. And Barbara McClintock, who was a geneticist from Cornell, she was studying Native American corn and trying to determine the genetics behind the pigment of the corn. And if you guys have seen Native American corn, it's beautiful, multiple colors, and often on one head of corn there'll be different colored kernels. She's trying to understand the genetics of this pigment production, and by intensive study of multiple generations of corn, she actually lived in the cornfields. She had a little field house, and she had a bed in there. She just lived there, and someone said she called the corn plants her children. She was a very dedicated scientist. She came up with a hypothesis that the only way that the changes in pigment could be understood was through these movable genetic elements that would insert themselves into the gene for the enzymes that would make pigment, knock out the genes for a while so that pigment wouldn't be produced. And then we now know that transposons can cut themselves out and then move to another site. So everybody said, you are crazy. You're a crazy woman. And it so often happens, folks that are thinking outside the box, folks that have a new proposal for kind of the worldview. Everybody says they're crazy, right? But luckily she hung in there, and it so often happens when people are humiliated for coming up with new ideas. She eventually won the Nobel Prize. So you guys, if people are laughing at you because you come with a new idea, hang in there. You too might get a Nobel Prize sometime in the future. So it is really hard when you come up with a new idea, and everybody says, oh, you're crazy. You're just bonkers. So what are these transposons? So I don't want you to worry too much about the structure, you guys, but just to try to describe this a little bit. The simplest transposon is called an insertion sequence. So this would be our double-stranded DNA of our simplest transposon. And transposons are characterized by what are called inverted repeats. And to me, this is always like, what? Okay, so an inverted repeat is, if we look at this strand and we look at the base sequence on this strand, it goes TAGTC over here. Okay, if we look at the opposite strand over here, kind of diagonally, we see the same base sequence is repeated here, TAGTC. So these are the inverted repeats that we see with all transposons. And then between the inverted repeats, the simplest transposon, they'll have a gene for the enzyme that permits the transposon to insert itself into one site and potentially either cut itself out or make a copy of itself and move to a new site. And the way the transposons target the sites that they're going to insert into is the target site will have these similar inverted repeats present. Now, some transposons, what they do is when they jump, quote unquote, what they do is they make a copy of themselves, and the copy gets inserted into the new site. So we'll have one copy of the transposon in the original site, and then a copy is inserted somewhere else. But in other transposons, it's been discovered that some transposons, they'll literally cut themselves out of where their initial location, and then they'll move to a new site. So there's different ways these transposons can jump. Now, as we mentioned, transposons can be important because when they insert into a new site, they might obviously they cause a mutation. There's going to be a change in the DNA base sequence there, and very often they'll knock out expression of a gene because maybe they cause a frame shift mutation, what have you. So they can be powerful in interrupting or altering gene expression. But another reason we're so worried about them is it turns out that there are complex transposons, and in addition to the transposase genes, they carry, of course, antibiotic resistance genes. And here again, we see another genetic element that can be transferred between bacteria that again can result in antibiotic resistance. So this is a transposon found in E. coli called transposon TN5, and it carries kanamycin resistance. And again, these transposons, they can make copies of themselves, move from chromosome to plasmids. The plasmids can be transferred to a new bacterium, and then the transposons can copy themselves again, insert into the chromosome. So we see not only the spread, but the amplification of these antibiotic resistance genes with the transposons. So, we see, bacteria have so many different ways to share their antibiotic resistance genes. It's just a cartoon of the transposition event. Okay, so just some little questions. All right, and this last little tidbit, you guys, that we're going to finish up with is a really brief discussion of the evolution of new genes, and we're going to focus on bacteria. And this concept of natural selection, how the environment selects those variants that are best adapted to their environment. And we want to apply this to the natural selection of antibiotic-resistant bacteria. And we want to see, again, we just want to prove to ourselves that us humans we play a big role in the selection of antibiotic-resistant bacteria. So usually the simplest way to show this, you guys, is to see, let's look at a population of microbes. And what we'll do is we'll use S to represent bacteria that are sensitive to some antibiotic. And we'll just call it a generic sensitive, I mean that the bacteria are either inhibited or killed if they're grown in the presence of antibiotic X. Okay, and as humans, that's what we want. If this bacteria is a pathogen, we want our antibiotics to kill them, right? And then we'll use a R to represent bacterium, or bacteria, which are resistant to antibiotic X. And that means that in the presence of antibiotic X, the resistant bacteria, they can still grow. They are not inhibited, nor are they killed by antibiotic X. And from, again, a human perspective, we don't want resistant bacteria, right? Especially if they're pathogens. So one of the questions in your textbook is, if we're talking about antibiotic resistance, and in general, genetic variation among the bacteria, one of the questions is, well, where do these antibiotic resistant genes, how did they initially evolve? How did they initially evolve? And you guys, you want to remember that good old DNA polymerase, even though it's very, it's a very good enzyme, it has proofreading and editing, we still have that low level of spontaneous mutations, right, mistakes that it makes. So the initial origin of all mutant genes is usually from mistakes that are made by DNA polymerase. So that would be the source of kind of our original genetic diversity. Even in our cells, you guys, the same thing's going on. Every time our chromosomal DNA is copied, our DNA polymerases will make, a few mistakes. So we've got a lot of genetic diversity in our own cells whenever they're copied. Okay, so let's say that we have a population of organisms, and just by chance, when one of the bacteria is ready to divide, DNA polymerase copies its DNA and makes a mistake, and it permits a change in an enzyme. So this enzyme perhaps can hydrolyze antibiotic X. It's just spontaneous, it's a coincidence, right? And a rare event, a very rare event, right? So most of the microbes in this population, most of them are sensitive to the antibiotic, right? And indeed, you guys, it's so interesting what microbiologists have observed, that in many populations of microbes, often, in environments where antibiotics are lacking, very often the resistant bacteria have lower survival rates because it often costs them energy, building blocks to maintain these resistance genes, to make the resistant gene products. It's wasteful if the antibiotic is not present, right? So one model is if antibiotics are not present, the resistant bacteria have lower survival rates. Now there's some experiments now that are challenging that. But anyway, we'll go with that for right now. Okay, so if there's no antibiotics present, the resistant bacterium has no survival advantage. And like we said, possibly these other guys even have a little survival advantage. They're not wasting time and energy making these antibiotic resistance enzymes that aren't required. So the resistant bacterium, low numbers, has a lot of competition here. It will remain in very, very low numbers, right? But when us humans, we come along, especially here in the United States, we love antibiotics, right? We love to ask our doctors for antibiotics. In medicine, we use a lot of antibiotics. And to me, this is going to be my plug, you guys. A place in the United States where we could really make big efforts to reduce antibiotic use is at least half our antibiotics go into animal feeds. Feed for cows and pigs and sheep and goats and chickens and turkeys. And the reason those low levels of antibiotics are added is it helps those animals grow faster, it helps them make more meat, it helps them make more milk, it helps them produce more eggs. So the antibiotics are not being used to treat a specific disease, it's being used to make the animals more productive. Okay? And again, over half our antibiotics in the United States goes into animal feed. Okay? So here in the United States, we live in this environment that's saturated with antibiotics, right? And so we'll just use the example of antibiotic X. Okay. Now we've changed the environment. Okay, we've got a lot of antibiotics X present. The environment selects the variant in the population that's best adapted to survive in this new environment. And which of our bacteria will be best adapted to survive in an environment that's rich in antibiotic X? Who's going to live? this guy, right? Everybody else is going to be killed. So what we see here is with our help, with our overuse of antibiotics, there's natural selection. The environment selects for the best adapted microbe, and in this case, it's the resistant microbe, right? So now all the sensitive microbes are killed, all the sensitive bacteria are killed. This lone survivor here has no competition for nutrients, for living space, is not inhibited by maybe toxic metabolic waste products of the neighbors. So what's this guy going to do? Replicate like crazy, right? So what can happen is we can actually get a change in the genetic makeup of our population of bacteria. We go from a population that initially was primarily sensitive to antibiotic. By using too much of the antibiotic, we have natural selection for the resistant bacteria, so that over time, we see a change in the genetic makeup of the population. Now we've selected for the resistant microbes. Now we've got a population of microbes that are totally resistant to the antibiotic. And even though this is oversimplifying, you guys, it's probably the basis of why we see now so many antibiotic-resistant pathogens. Yeah, is our overuse of antibiotics, inappropriate use of antibiotics. And as I say, I think that one place in the United States we could make huge progress in reducing antibiotic X is that if we could decrease the use of antibiotics in animal feeds. And the problem is, and I know this is always a big issue, is that it'll probably increase the cost of milk, meat, eggs, poultry, right? But gosh, you guys, we're getting to the point now, some of our pathogens are becoming resistant to almost all of our antibiotics. So it seems like somewhere along the way we've got to put a break on this. That's just my little spiel. And I know I shouldn't be up here on my soapbox, but that's just my okay. So it is important that we understand how humans are involved in creating the natural selection for these antibiotic-resistant bacteria. Now, another thing I'd like you guys to know for exam 3 is how is it that bacteria can survive in the presence of antibiotics? What are the mechanisms of antibiotic resistance? So we just want to go over that, make a little list here of some of the ways that bacteria can survive in the presence of antibiotics. Okay, so, and we're going to start by this because this is one I definitely would like you to know for exam. Maybe be on the short answer section, I'm not sure yet, but really want to make sure you guys understand this. So, mechanisms of antibiotic resistance. And when I say mechanism, I mean, exactly at the molecular level, what is happening that permits bacteria to grow in the presence of antibiotics. So we'll first start with some general ideas, kind of almost non-specific resistant mechanisms, and then we'll go to the more specific mechanisms. Okay. So first of all, cell structure may play a role. So what do I mean by cell structure? Well, let's think of bacterial cell walls. We know there's 3 different types of bacterial cell walls. Gram-positive cell walls that have thick layers of peptidoglycan. Peptidoglycan is very hydrophilic. It does not provide any protection against antibiotics. In contrast, we have the gram-negative cell wall that has that hydrophobic outer membrane, which is definitely a protective layer against many antibiotics. It will inhibit the diffusion of large hydrophilic molecules across the outer membrane to reach the peptidoglycan or into the cell. And then, so let's put that gram-negative outer membrane of the cell wall is protected. And you guys remember one example is that many gram-negative bacteria are naturally resistant to penicillin because penicillin can't cross the outer membrane. You'll recall that if we have large hydrophilic antibiotics that we're going to use against gram-negatives, the antibiotic must be able to pass through the porins, those protein channels in the outer membrane. Okay, so penicillin, as an example, third type. The acid-fast, right? The acid-fast cell wall. And this is the most protected, the most hydrophobic cell wall of bacteria. We want to remember the acid-fast bacteria, the Mycobacterium, Mycobacterium tuberculosis, Mycobacterium leprae. They have these incredible hydrophobic, waxy cell walls. And indeed, that is one of the most protective type of bacterial cell walls. So many, many antibiotics cannot cross that acid-fast cell wall of the mycobacteria. So the mycobacteria are naturally resistant to many antibiotics. Another cell structure of importance are endospores. Are endospores destroyed by antibiotics? You could probably soak endospores in concentrated antibiotic, and they're just like, right? So we want to remember the endospore, it's incredibly resistant. It is impermeable to most chemicals. So the antibiotics are not going to destroy the endospore. There will be no damage to the DNA that the endospore is protecting. So we need to remember that. This becomes important, and I think we might have mentioned this, you guys, in cases of inhalation anthrax, there's a lot of concern that some of those endospores remain dormant in our lungs, maybe for more than 2 months. So traditionally, when we had the anthrax spore being shipped through the mail, folks were put on Cipro for like 2 months. And now physicians are wondering, wow, maybe we should have extended that, because possibly some of those endospores don't germinate for a long, long time. And so if you take the patient off, the antibiotic and the endospores are still present, they germinate, then the patient could develop inhalation anthrax. So you gotta remember those doggone endospores. Yeah. Okay, so that's based on cell structure. In this cartoon, this is out of the antibiotic chapter in your textbook, chapter 20. This is figure 20.20 on page 575. This first mechanism here, blocking entry. This specific examples would be the gram-negative bacterial cell wall, the acid-fast cell wall, and endospores. Okay, so we're going to block entry. Now, another behavior that bacteria have evolved that will protect them non-specifically against many different antibiotics is the formation of biofilms. And you guys recall that folks are thinking that now perhaps the majority of bacterial infections in humans and animals, perhaps in the majority of those infections, the invading pathogens are forming biofilms. And you guys remember that a biofilm is a community of microbes that forms on some surface. Okay, so artificial surfaces like artificial hip joints, indwelling catheters, pacemakers, artificial heart valves are great places. Also, any kind of damaged tissue in the body is a great place for biofilms to be formed. And you guys remember a biofilm is a community of microbes, which I'm indicating just by these little dots. A community of microbes that live in what's called the sticky extracellular matrix. This is usually rich in polysaccharides. It's the slime layer that these microbes produce. And you'll also recall that very often, since it takes a long time for nutrients to diffuse through these layers, often the microbes, at least in the lower layers, are in the stationary phase of growth. And we remember from chapter 6 and the standard microbial growth curve, bacteria are most sensitive to antibiotics in which phase? The log or exponential phase, right? So in stationary phase, they aren't growing very fast. They're not metabolically very active, and thus they're less sensitive to antibiotics. So bacteria living in biofilms, then, since they're in kind of this resting phase, they're just inherently less sensitive to antibiotics. And also, you want to recognize that the layers and layers of slime and microbes here, that too creates a diffusion barrier, right? So the microbes in the deeper layers here, very low concentrations of antibiotics are going to be established in these deep layers. So the biofilm itself is a physical protective barrier against many of these antibiotics. Okay? And we described that this is perhaps a reason why a lot of folks, when they get treated with antibiotics, the bacteria can't be killed. So often they have to go in and surgically remove, if artificial surface, they have to go in and remove it. If it's like maybe damaged heart valves, they have to go in and maybe do, valve replacement surgery. So sometimes the bacteria are so resistant, they have to go in and somehow get rid of the biofilm before the infection can be successfully treated. Okay, so another way that bacteria can resist antibiotics is through enzymes which either hydrolyze the antibiotic, and our example were the beta-lactamases that we talked about that most Staph aureus produce that can destroy penicillin, ampicillin, amoxicillin, and in addition, bacteria might have enzymes which chemically modify. So I'll put or chemically modify the antibiotic. So for example, some bacteria have enzymes that they'll add an acetyl group to antibiotics. And when you add that additional chemical group, the antibiotic can no longer bind to its target. So you have chemically modified antibiotics, so antibiotics can't bind the target. Okay, so that's another common antibiotic resistance related enzyme. There are these enzymes that chemically modify the antibiotic, so then they can't bind to the target. Okay, so here's another mechanism. You can have mutations in the bacterial genes for the target of the antibiotic, so that you get a change in the shape of the target, and thus the antibiotic can no longer bind. And this is often seen, for example, we had shared with you that there's an antibiotic called rifampin that inhibits bacterial RNA polymerases. Well, rifampin is rarely given as a single antibiotic because all it takes is a point mutation in the bacterium, and the RNA polymerase is changed in structure so that the rifampin can no longer bind. So in that case, whenever you have antibiotics to which bacteria rapidly become resistant, you always give an antibiotic in combination therapy. You always add another antibiotic or maybe 2 or 3 other antibiotics. Okay, so that's an example where through mutation, the bacteria changes the structure of the target so that the antibiotic can no longer bind. Now, this last one here, these efflux, these are called efflux pumps. They're protein pumps that will literally bind to the antibiotic within the cytoplasm of the bacterium and then pump it right back out of the cell again. These are really scary. I didn't realize this, but some of these antibiotic efflux pumps are very nonspecific, and that means they can pump a wide range of antibiotics out of the cell. So this is a huge concern. One of the big debates over one of the initial antimicrobials that they were incorporating into toys and soaps and cutting boards, it was called triclosan. And what they discovered was that overuse of triclosan selected for bacteria that had these nonspecific antibiotic efflux pumps. So not only were you selecting for triclosan resistance, you were co-selecting for resistance against antibiotics. The triclosan is considered like an antiseptic disinfectant. Okay, but by selecting for these triclosan pumps, they were pumping the triclosan out of the cell. Those pumps were also able to pump other antibiotics out. So it's like, wow, we don't want to do that, that's for sure. Okay, all right guys, so do remember, maybe as a short answer or some multiple choice questions, I'll ask you for the mechanisms by which the bacteria can resist these antibiotics. How is it that they can live in the presence of these antibiotics? And because we're running behind you guys, we're going to skip this one. This stuff is so juicy, I can't stand it. Okay, this is really quick, really quick. So this is monitoring the presence of antibiotic-resistant bacteria in a poor patient that was suffering from a chronic kidney infection caused by gram-negative bacteria. So here in black is the number of bacteria that were present, and I'm presuming in the perhaps a milliliter of blood, maybe we had a little septicemia going on. And then this represents the antibiotic resistance. And this is telling us how many milligrams per mil of blood, for example, was required to kill the bacteria. And what we see here is that initially at the start of therapy, we had high numbers of bacteria per mil. They started the antibiotic therapy at low levels, like 1 mg per mil. And we can see there's a dramatic death rate here. So most of the bacteria died. But what happened here, you guys? Yeah, there's two possibilities. We either had a spontaneous mutant in here that became resistant to streptomycin. The drug that they were using was streptomycin. This is an aminoglycoside. It targets the 70S ribosomes. And so sometimes you can get mutations in the genes for the ribosomal proteins or ribosomal RNA so that the antibiotics can no longer bind. Maybe it was a spontaneous mutation here. Or it's possible that in the initial population, there were a few naturally resistant bacteria, just like we drew on the board. So what was happening here is you were killing all those sensitive bacteria potentially, and who survived? The resistant one. And now they don't have any competition from their neighbors, so now they start multiplying like crazy. So now this is where you have that natural selection for the resistant bacteria. Most of the pathogens now are streptomycin resistant. And you can see what they did here is they saw that the bacteria were no longer responding. So they just, oh my gosh, they just went bananas, increasing the dose of the streptomycin. And at least they got it to level off here. But what's so sad about this, you guys, is streptomycin as an aminoglycoside, it's nephrotoxic. It damages kidneys. And I'm like, gosh, this poor person, they already had kidney damage from this infection, and then they're giving an antibiotic that causes further kidney damage. So this is like, well, it's like, okay, we might have killed the bacteria, but I wonder if the patient survived. It's like, wow. So you gotta, whoo, boy, that's a tricky one. Okay, so that's that one. And this, you guys, please do read this. There will probably be a question on the exam. This is a clinical focus. It's on page 577. Again, it's in the antibiotics section of the book, but very important. And this is, and I promise I'll be quiet about this after this, you guys. This is an example of how incorporation of low levels of antibiotics in animal feed, in this case in poultry feed. Poultry is like chickens, turkeys, ducks. Incorporating low levels of antibiotics in poultry feed, and again, this was done to increase growth. So you have heavier chickens slaughtered. It's used to increase egg production. Okay? What we want to see here is that when fluoroquinolones, for example, the equivalent of ciprofloxacin, was incorporated in low level in poultry feeds, we saw an increase in the evolution of antibiotic resistance among a very important group of foodborne pathogens, the Campylobacter. Okay? So let's just review that Campylobacter. In chapter 6, we talked about Campylobacter as being common contaminants of poultry, right? Mm-hmm. Chickens, just figure, you guys, whenever you buy raw chicken, just presume it's got Campylobacter growing on it. In biofilms, remember? Mm-hmm. The Campylobacter, they're microaerophiles, so they form biofilms, and they'll have just the right concentration of oxygen to grow in that biofilm, right? And we said Campylobacter can cause foodborne illness, vomiting, nausea, diarrhea. And a huge concern is that some folks, and it's probably genetically determined, some folks, after being infected with Campylobacter, they develop an autoimmune disease where their immune system attacks their nerve cells and neurons. And this causes Guillain-Barré syndrome. You have a paralysis of the limbs. Now, in a lot of folks, it will eventually recede, so you don't have permanent damage. But in some folks, it can cause permanent damage, and in even some folks, it can kill them because they have paralysis of the muscles involved in respiration. So Campylobacter infections, we want to respect them, right? Okay, so why do we worry? Well, let's take a look here at the history of the use of fluoroquinolones, for example, ciprofloxacin and its relatives. So fluoroquinolones were first introduced into human medicine in the late 1980s, right? We weren't using it in vet medicine nor in agriculture yet. And what we can see is within about 3 to 4 years, we start seeing these fluoroquinolone resistant Campylobacter jejuni isolates from humans. So we're getting some resistance. Okay, it makes sense. But let's say if we look at the slope, if the slope for Campylobacter resistance was even, okay, it should have been about like that. But suddenly it takes off like that. And what happened? This is the year when fluoroquinolones were incorporated into chicken feed. So we've increased the amount of fluoroquinolones in the environment. We're increasing selection for those resistant bacteria. And so if these represent an increase in the number of Campylobacter jejuni resistant fluoroquinolones from humans, what this is telling us, you guys, is we're probably getting transfer of resistant Campylobacter from those chicken carcasses that were fed the fluoroquinolones. We're getting transferred to humans. And those of you that have ever worked with raw chicken in your kitchen, especially chicken carcass, and you're trying to cut it up, if I get my cleaver out and I'm like, whack, 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 I mean, there's juice, and bits of tissue flying everywhere, right? So it's really easy to contaminate your kitchen with Campylobacter from a chicken carcass. So just be careful, you guys. Now, what the good news is, and here in the United States this is so hard to do, is when it was recognized that the fluoroquinolones in the poultry feeds were probably increasing the rate of fluoroquinolone resistance in Campylobacter, they finally discontinued its use in the poultry. And what we're hoping is that we'll see a reduction in the number of fluoroquinolone resistant Campylobacter. Okay, so yeah, this overuse of antibiotics, especially in agriculture, gosh, this is a place we could make real progress, I think, in the United States, and I think it would really help. Yeah.
So even though we might see a reduction in the resistant gene, when they die off, they might release their DNA, and the S strain might accept that.
You are, oh my gosh, you just nailed it. Yeah, because there's a hot topic now that, okay, you know how we're always worried about bacterial contamination? Like, we're talking about in the lab, fecal contamination of food and drinking water because we're worried that intestinal fecal pathogens could be present. So always before, our focus was on how water and food would be contaminated with living pathogens, right? Well, now a hot topic is DNA contamination of the environment. And one of the papers, right, and you guys, oh my goodness, after this class we say everybody just wants to hide under their covers in bed. So it's exactly what you're saying. So let's say we've got a farm and they're feeding lots of antibiotics, say to their dairy cattle, to increase milk production, to the dairy cows, to increase milk production. And this is, this is not that unusual. So what happens is on a lot of these big farms is the manure that the cattle produce, the manure is going to be chock full of bacteria, right? And because you're giving them antibiotics, you're probably selecting for antibiotic resistant bacteria. Okay. And then what a lot of these big farms do is they'll harvest the manure and they'll use it for what? They'll use it to fertilize next year's alfalfa crop. Alfalfa is harvested and made into alfalfa